Namaskar dosto, welcome to channel Toxicology on board. Today in this video we will be discussing another DABT question. The topic will be lead mode of excretion in human adults. In my channel I have already uploaded certain videos with respect to lead. So always these metals, if you look into lead, arsenic, mercury, they are not enough. No matter how much we read, there will be always additional information that is needed that might be a topic of question for the DABT questionnaire. So let's see what important things in this video we can learn lead with respect to kinetics or its excretion. Okay, how it is different, the excretion, how it is different in animals as well as that is in the particular in the case of rats as well as in the human adults or in the infants like that or susceptible population. So let's directly dive into the question. This question is taken from reference ABT recertification examination May 2000. This particular question paper from this I have taken this question. So upcoming whatever few couple of next videos will be from this exam question paper only. So the question was the dominant route of excretion for absorbed lead in adult human is. So we have to identify what is the route of excretion, particularly the year the age of the population is adult, that is in human. So option A is bile, option B is feces, option C is urine, option D is epithelial tissue, option E is respiratory tract. The right answer will be in the green text, that is option C, urine. So with this question, we will also try to understand in detail about lead, its kinetics, and important points that we can remember with respect to lead. See, if you once a person, adult, consumes lead, no, the it is mainly once it enters into the human body, it is excreted mainly through urine and feces. So the primary route of mode of excretion for this metal, lead metal, is urine and feces. The right answer was C. You should not get confused. Even in the option B is there, feces. You should not take the right answer will be option C. Okay. There are bile, epithelial tissue, respiratory tract. They are not the right answer, but a small amount of lead can also be excreted through sweat, through hair and nail. But this process is no, it is too slow. Okay, and some part of the lead once you take is ingested, no, it will go and deposit in your bone. Okay, so especially from the bone stores, with the elimination from blood happening faster, because there is a whatever the concentration plasma levels in the of the lead in the plasma increases, no, it is the excretion is faster through this route, the around 50 to 60% in weeks, but skeletal lead can take up to years. Okay, once the lead goes and deposited in the bone, no, it will be there attached to this bone for couple of years. Okay, it is not completely eliminated from your body. Okay, so important things that we can learn with respect to lead, these next important two slides have been taken from Clayson notes only. Okay, there is a subject called Clayson short notes. From this only I have kept. So if you look into metal, no, there are few, as I told you in the beginning of the video, important metals like mercury, arsenic, lead. These things, so you have to remember. One or two questions you will be definitely getting in exam. So lead is similarly lead. Why these certain metals are so much important means that because of the nature of the abundance in the environment or the surrounding through which a human can get exposed. That is the reason lead is ubiquitous in nature. Okay, the most susceptible are infants and fetus. Now you have to understand which of the following population is more susceptible, adult or infants or fetus. Adult, when you compare all these three type of populations, no, children as well as infants and fetuses are more susceptible to the toxicity of lead. The reason is more GI absorption and certain systems are not yet developed in this infants and fetus. Major reason is the GI absorption rate is faster. If, for example, 100 mg lead is there, if it is given to adult as well as infants and children, majority more absorption will be there in the case of children or infants when compared to adult. Absorb much higher percentage than adults. Exposure through food and water. If you see the route of the mode of exposure, lead, it is more if the food as well as your water is contaminated with lead, the exposure is through this is more when compared to air. Occupational exposure through air, you can say it is lesser. Lead, 90% of the lead in blood in RBC, membrane and hemoglobin in equilibrium plasma. This another point we'll discuss. Actually what is happening, lead will go and bind to this particular membrane of hemoglobin, so that we will see one more sentence will be there, I'll discuss. So lead is also found in bones, as I I told you, once it goes and deposited in the bone, largest pull and slowest turnover, since it is bound to the bone. Also in the CNS, you can see that is gray matter, hippocampus. Hippocampus is more than cerebellum or cerebellar, cortex and medulla. This is the region where you can see the pool of the lead getting deposited. Excretion in the urine, as we saw in the question also, the major route of excretion is urine, but certain times tubular reabsorption of this lead can also takes place. Coming to toxicities, major important toxic effects, adverse effects or toxic effects of lead are various organ systems it will be affecting. One is nervous system. Nervous system, particularly in the case of infants, what is happening in the case of infants, blood-brain barrier is not developed completely. As a result, infants are more susceptible to this nervous system damage with exposure to lead, because of which what is happening, encephalopathy, swelling of the brain takes place, and children might undergo blindness, mental retardation, and low IQ will be there. Okay. Uh, how it is affecting peripheral neuropathy, lead is main important thing. This is the classic feature. It causes foot and wrist drop. Here you can see in the figure. Okay, this is the thing it causes, and it causes segmental demyelination. Myelinated neurons, demyelinated neurons will be there. Schwann cells will be there, where the transmission of signals takes place. Jumping signals will be taking place. No, that particular segment demyelination is taking place because of the lead exposure, and motor neuron nerves more sensitive than sensory neuron. Since we have two types of neurons, motor neurons and sensory neurons, motor neurons are more susceptible to this lead toxicity. How the adverse effects of this lead on kidney, it causes nephropathy, particularly which part of the kidney, renal system, that is kidney, nephron, tubular, it is particularly damaging proximal tubule inclusion. So it is affecting the proximal tubule. When you perform histopathology of this, no, you can see inclusion bodies, you can see. Okay, and lead protein complexes will be formed in the case of PCT, and hypertension. There is a elevated, whenever there is an exposure to lead, there is an increase in blood pressure. That is the most sensitive adverse effects from the lead. But this can't be the classical feature, because there are certain other chemicals also when we are exposed, it can increase the BP, but this can't be the case. But there is a common most sensitive thing you can see. Lead can substitute for calcium. If lead is there, calcium is there, and there is a neurotransmitter release is there across the neurons, what is happening though? Lead will substitute in place of calcium. In that neuronal junction, no, calcium should be released. Instead of calcium, lead will be released. Okay, because of which what happens, it will interfere with this calmodulin dependent reactions. As a result, it will inhibit Na plus K plus ATPase and interferes with the energy metabolism. So whatever the amount of ATP energy needed during this Na sodium and potassium ATP exchange, that won't happen, because lead is substituted in place of calcium in the neurotransmitter release. Next one is, how is the adverse effects of lead on the on the blood. If you see, there are different types of anemias will be there, particularly because of the lead. What is happening? Anemia shortened the lifespan of the RBC. Okay, RBC life number of days will be decreased. Low heme formation, iron deficiency, inhibition of this very important one. Aminolavulinic acid dehydrase. This question you will get. Aminolavulinic acid in the urine serve as the Burley biomarker of lead exposure. If a person is exposed to lead, and if you perform a urine analysis, and if you find this aminolavulinic acid in the urine, it will serve as a biomarker to identify, yes, that is caused the toxicity in this particular individual. Then what are the adverse effects of lead on the reproductive system? It is affecting the sterility, and it is affecting the decrease in the sperm count in the case of male reproductive system. How the is lead is causing cancer or not? There is no classification given by IARC, or but it is told, it is observed that lead is a probable carcinogen, particularly digestive and respiratory tract, but not kidney in the case of humans. If the lead was tested in the rat 2-year carcinogenicity study, they have found that there is a formation of renal adenocarcinoma as well as tuberous hyperplasia was observed. Okay, same things were not observed in the case of adult human beings. Another very very important thing with respect to lead you have to remember is Burton's lines purple coloration in the gingiva. So this is the another way where you can a physician can identify that there is a lead toxicity to certain individual, that is Burton's line purple coloration in the case of gingiva. And how to mitigate the risk by using chelating agents like EDTA, BAL, DMSA, these which are being FDA licensed. Okay, so we will read in some more additional points with respect to lead which were missed in that previous slide. As no major source of contamination for the lead are fuels soldered from tin cans. Those workers who are working with this soldering tin cans, the tin cans which in which food is stored, and in that if there is a lead is leaching from the tin cans into the food product, that is a source of contamination. Pottery, there are certain pottery clay utensils in which you will be cooking your food, but if they are glazed, at that time also there is a source of contamination, exposure. And smelters also, those who are working on the welding, everything, no, those also. Next, so as we discussed earlier also, lead, the exposure is more through ingestion than from food and water, but not through air. Occupational exposure through air, it is not there. uh, this is very again important. Lead, it is highly more absorption is through GI absorption in the case of children and infants than compared to adults. And this is again very important point that you have to remember. The lead retention by the body is cumulative. So there are certain questions where the DABT examiner will ask you with respect to synergistic effect, additive effect. So like that also, the one question in the option you might be getting about cumulative. So this lead, it is causing cumulative toxicity, lead retention by the body. For example, 100 mg lead you have given on day one, and on day two again your repeated exposure is taking, but there is a cumulative, there is a residue of the first exposure still there in the body. There is then on the second day additional dose will be there, cumulative toxicity will be there. Okay, then important thing you have to remember about, more than 90% of lead in the blood is bound to RBC. I told you, no, we'll learn about this one. The RBC may be also, it is bound to this membrane or a hemoglobin. Okay, and blood lead levels are all used for, yes, so this is used for, particularly used for risk assessors, risk assessment के लिए in the water bodies if lead is there, for that type of no exposure assessment, risk assessment के लिए this is very important. The levels of the blood in the levels of lead in the blood, it will be asked as a risk assessment tool, I mean to say. So another very very important point with respect to that question also you have to remember, there are two types of kinetic pools are there with respect to lead exposure. One is skeletal pool, which is the largest, and lead in this pool has half life of 20 years. For example, today I am getting exposed to 100 mg of lead. If it is that 100 mg, for example, 60 mg or 30 mg, 40 mg is going and deposited in the bone, it means it will be there in our body. Its the half life is around 20 years. So this is about the skeletal pool. Second one is soft tissue pool, which is much more liable. So this is with respect to plasma that can be easily excreted through either urine or feces. Okay, lead produces a wide range of adverse effects that we'll see. As we told, no, in the children it is definitely affecting your nervous system, and up to 50 to 70 micrograms of lead in the blood level, it shows no clinical symptoms, but still it will affect their IQ of the children will be affected there. So psychomotor or developmental test of this particular infants, no, it is very less. Coming to high doses, in the case of high doses, no, the children will show lead encephalopathy. As I told you, there is a edema, severe edema of the brain will be there. Lead can inhibit the cholinergic function, possibly be reduction of this extracellular calcium that we told you. Lead is, instead of calcium releasing into the neurotransmitter release, lead is getting released into the neuromuscular junction. Then lead can inhibit dopamine uptake is inhibited, function of neurotransmitter GABA is inhibited. Okay, and classic signs of lead toxicity, wrist and foot drop. This is again you have to remember, Burtonian lines of wrist and foot drop. Children may, infant may encephalopathy. These are the very repeated questions you will be getting. Segmental demyelination, possible axonal degeneration may also follow Schwann cell degeneration. I told you, no, myelin sheath will be there and that will be disrupted, because of which transmission of signals will be decreased. As a result, I think the previous slide with the psychomotor functions, those will be inhibited. Lead induced anemia, types of anemia you can see, microcytic anemia it is causing, hypochromic anemia it is causing, which is similar to that of in the case of iron deficiency, and it produces basophilic stippling of reticulocytes. If you see, this is again a classic feature. So which of the following will cause basophilic stippling of reticulocytes? Then you have to see if the options are the arsenic, lead, mercury, so and so. You have to select lead. Which of the following causes Burtonian lens? Answer is lead. Like that you have to remember. RBC shows a shorter lifespan. Yes, the one who are exposed to the lead node, the lifespan of this RBC is decreased. Okay, possibility to increase membrane fragility. Why? Because this lead is going and binding to the RBC membrane, hemoglobin membrane. Because of this, that membrane is getting fragile, and the RBC will die before only. Okay, and heme synthesis is impaired, probably because of this is again important. Why new heme or new RBCs are being interrupted, their number is decreased because of this delta amino levulinic acid dehydrogenase inhibited. This particular enzyme is inhibited because of this heme, which is very important in the case of heme hemoglobin formation, heme formation. Then ferrocatalase, another enzyme is there. The insertion ferrous iron into porphyrin ring, and that can reduce this ferrocatalase activity. Another enzyme it is inhibiting, that is ferrocatalase activity, leading to porphyria and gouty nephropathy. Okay, again you have to remember. And several genetic polymorphisms are also include common in the population. These may relate to individual susceptibility because of the genetic polymorphism variation. The reaction to the exposure of this particular lead will also differ. In the kidney, lead poisoning is the presence of nuclear inclusion bodies. The previous picture I showed you, nuclear inclusion bodies are nothing but they are very early signs of lead exposure. These bodies help in lead excretion through the urine. Lead affects mitochondrial respiration as well as phosphorylation, because of which ATP is getting inhibited. There are reversible tubular effects, irreversible chronic interstitial nephropathy, causes amino aciduria, glucosuria, phosphaturia, but no loss of calcium is taking place. That appears to increase the elevated blood pressure we have seen. This is the most sensitive adverse effect in the case of general population. Whether it is carcinogen in animals, yes, in the rat we can see adenocarcinoma was there. Okay, but there is no enough data in human to conclude that this particular lead is carcinogenic. Okay, important thing again, Burton lines, purple blue color discoloration in the gingiva. It's a classic feature in the case of children with a lead encephalopathy, and the treatment is either through EDTA, chelating agents, or BAL, or a combination of this. So hope from this video you were able to learn about lead toxicity, particularly with respect to, in this video we have learnt about the kinetics and important target organ toxicity which lead is causing. Okay, and important route of excretion, and why there is a change in the skeletal pool with respect to adults, human. Okay, the sensitive population. So thank you so much. Be curious. For any questions, please comment, or you can write a mail to arya.priyanka18@gmail.com. Like, subscribe, and follow my YouTube channel Toxicology on Board. All the best for your exam. Thank you.